it was not really planned to organize it in seasons. I mean, I literally came up with it 15 seconds ago or something because after last year I struggled to keep things going with this program, especially since after I moved to Denmark, where I am currently working as a research assistant, and I've been mostly caught up with that, which hasn't changed, but still, definitely wanted to revive this program for a long while now. So I decided to try and go and give it another go. And I hope you still remember me. At least someone out there still remembers the interviews that I was conducting back last year. And also, of course, since Haps, this wonderful platform that we're using, allows you to support creators and streamers with those coins that the platform itself gives you. So don't forget, I would really appreciate it if you would be so kind to toss a coin to your snake man, so to say, to keep it in the Netflix series um, vibe, so to say. Yeah, and so now I've already enough with my babbling, and let's go straight to introducing our guest of the day. Now, I've known him for a couple of years. By now, I met him back in Australia when I was doing my master's thesis at Brian Fry's lab at the University of Queensland studying snake venom. And he was already a PhD student there, working on venom as well, but on a crazy variety of different organisms, from snakes to yeah, insects. I believe at some point as well, invertebrates. At least it's been a while. But perhaps most interestingly, fish, because yeah, fishes can be venomous too, and I believe there is no one more qualified to discuss that than our guest of today. So join me, everyone, in giving the warmest of welcomes to Richard Harris, who will be joining us live in a few seconds. Hey, Rich, can you hear me?
Hi, hi Lorenzo. Yeah, can you hear me?
Oh yeah, loud and clear.
Yeah, brilliant. Excellent.
Perfect. So thank you so much for joining us today. Welcome to the Snake Man's Lair.
Nice, nice. I'm excited. Yeah, this
Yeah,
is a first for
I
me.
already
So
I already know that either in your mind you're either thinking that the title is cool as hell or thinking that it's the most stupid thing ever. So it could
no,
be
look,
either one
it's
of those.
fish venom's my thing. So fish venom's my thing. So I'm happy with any title.
Good to hear. So yeah, as I was saying, you are well, actually, I would dare say one of the world's leading experts when it comes to fish venoms. Uh, and
Well, I think that's probably because there's very few people.
yeah, but it still counts. I mean, man,
Yeah, I
you gotta
guess.
sell yourself. Come on. uh,
There's a slight echo, someone said.
oh yeah, apparently.
Uh,
So let's turn this echo off. Yeah, I turn off my echo cancellation. I hope it's better now, David. So yeah, let us know if it's still there. But yeah, so I mean, I was definitely excited to have you here, especially for your work on fish venoms, because again, like as you know even better than I do, when you say venom, like people mostly think of, well, snakes, of course, like so
yeah,
scorpions or spiders, perhaps even some insects, but definitely not fish, or at least not
no,
that I would imagine. So
yeah.
how common is it in fish, like to have venom? Like how many, what proportions are we talking about here?
Okay, so yeah, I mean, as you say, like everyone thinks of snakes and spiders and scorpions when they think of venom, right? And they don't actually think that a lot of fish are venomous. But if I can just kind of put it into perspective, so if we take every single species of vertebrate, for example, 55% of vertebrates is fish. Okay, so we have this huge diversity of animals already within the class of vertebrata, right? So 55%, that's an insane number. That's more species than amphibians, reptiles, mammals, birds put together. Okay, and so when you have so many different, species and such a variety, it's no wonder that adaptations arise. Okay, so one of these major adaptations that people don't realize is venom. Okay, and so to put it into perspective, within fish alone, because they're very understudied in terms of their venom classes, there's estimated to be about 2,000 different species of venomous fish. And yet, if you asked one person to name 10, they could probably name you one or two, maybe the lionfish or the stonefish, for example, right? And so we have this huge understudied diversity of venomous fish that, uh, due to many different reasons, why people are not really researching them. But there's just a huge scope. And again, to give another kind of statistic, I guess venoms have risen about convergently evolved about 19 different times within fish. So that's an insane amount of convergent evolution, right? On this one system, this defensive or in some cases predatory system. And so, yeah, so if it's convergently evolved on 19 separate occasions, that means there's a huge, selection pressure for fish to actually evolve these systems. And it seems relatively, I don't want to use the term simple, but to arise that many times, it seems like, you know, there's clearly something going on that we're not really investigating, right?
No, yeah, I mean,
So I think, yeah,
I definitely didn't know that it arose 19 times
yeah,
independently in fishes alone. Like,
yeah.
because I was just like maybe tiny little bit above the stonefish and lionfish, and then that's it level. But still, this kind of blow my mind, I must admit.
Yeah,
So
and
yeah,
even
but
even within major groups. So, for example, your scorpion fish, for example, it's thought to have evolved at least one or two times in there. Catfish, for example, are probably the group within the fishes that's evolved independently on many separate occasions. I think it's like four or five times within siluriform catfishes alone that it's independently convergently evolved.
yeah, so even catfishes.
Mm. Yeah.
So
And even the
what
ones in the pet trade as well. So Corydorus catfish, for example, that in the UK particularly, you could literally go down your local pet store and buy as many Corydorus catfish as you like. And so yeah, again, it's like a lot of people are unaware that they're venomous. And um, one interesting thing about the Corydorus catfish example is the fact that they actually have a lot of mimicry systems as well that arise with the venom. And it's just so interesting to me that, because the fish trade, for example, the aquatics trade is so common, and yet a lot of people are so unaware that even some of the fish that they just pick up in their marine tanks or freshwater tanks might actually be venomous and potentially are.
Yeah, so yeah, even those catfishes, like now I'm wondering what do they use the venom for? Because like you mentioned, of course, predation and defense, and there are the famous cases like the lionfish that won't kill you but will make you wish you were dead. Um,
Yeah.
but yeah, aside from that, like what kind of like physio biological ecological uses do we see in fishes when it comes to venom?
So yeah, so the majority of venomous fish use theirs as defense, because they already have these, like a lot of spiny ray fish, for example, have already that venomous spines in place where venom can evolve or venom can adapt to that area, so it makes it easier. And these are obviously defensive adaptations. They stop them from being predated, and they cause a lot of pain. Like some of the descriptions of people being stung by venomous fish just describe it as the worst pain imaginable. And as you mentioned, then it's like you'd rather wish you were dead than go through the agony of some of the pain. So yeah, so the majority of fish venoms are evolved for defensive purposes, but there are some instances where they have evolved for predatory purposes, or in some cases potentially competition. So as a predatory purpose, you think of something like a lamprey. So they basically just kind of attach to their host. They produce anticoagulants within their venom, and some of them have produced chitinases too, which kind of break down the skin layer, the chitinous fish scales just to kind of predate that blood. Then you have other fish, for example, the fang blennies, which are some of my favorite example of venomous fish. Um, and so essentially what these do is these kind of predate on the scales of very larger fish. But the venom's actually really cool because it actually induces an opioid response. So it kind of makes the whatever they bite, it kind of makes them, in a sense, high or stoned, which is really cool. So it disorientates the other fish that it bites, for example. And so this is great actually, and it's thought that this might actually be a competitive thing between local species because if you think about it, right, if you can induce kind of this disorientation with your nearest competitor, then they're not going to be able to swim properly, and then they're going to be taken up by a larger predator. So once they're removed from the local space, that frees up that kind of niche, less competitive area for you to just explore, right? And so it's a really cool example of a venom system that could potentially have been evolved for competitive purposes. But as you say, the majority of fish venoms are defensive, like hell.
No, but I see why you like the Blenny so much because like
Yeah,
this is kind of crazy if you think about it that evolution would even come up with something like that. Like
yeah, it's crazy.
you make your own conspecifics hide and stoned to make them to facilitate them being eaten by something else.
Yeah, it's
That is
really really cool adaptation.
even when saying it, it sounds absurd, and yet it's out there.
Yeah, it is so cool, and I think com Venom evolved for competition is very very rare within the animal kingdom alone. So you have the example of the platypus, the male platypuses,
Yeah.
for example. Um, but there's very few instances where that has arisen, and so again, that's a very cool adaptation for fish.
Oh yeah, David is asking, is that the same little blenny I find in rock pools? I wouldn't know. Maybe.
I no, so these are coral reef. So yeah, I don't think it'll be the same species. So the species that this is Mycanthus grammistes, so part of the Mycanthus genus, the fang blennies. I mean, it's possibly you find them, or at least some relatives within rock pools, but I'm not 100% sure. They're mostly coral reef dwelling. So yeah.
Yeah, and speaking of getting high, like one of your most recent papers was aptly titled "Getting Stoned," but
Okay.
this time for a completely different reason because it was about stonefish, which are kind of the cash cows when it comes to
Mm,
venomous fish, so to
most
say, because
definitely.
they are also those of the with highest clinical relevance, I think, and. So about that, in that paper, you examined stonefish venom and you tested its toxic effects on semi-physiological, on a sort of a replica of a physiological system. And so, what did you see in there that was so interesting, and why is it relevant in medical terms as well?
Yeah. Okay. So firstly, we tested we wanted to test the venom on a few kind of different in vitro assays. One of them was looking at how the venom targeted nicotinic receptors. And so, for anyone who doesn't really know, nicotinic receptors are um, receptors. The ones we were looking at were the ones located at the neuromuscular junction, so they allow muscles to contract and things like this. And so there was evidence kind of maybe 10, 15 years ago that the venom actually potentially targeted this area, but it wasn't conclusive. And so we wanted to follow up on this. And so I tested the venom on an assay that I've been working on for my PhD, which essentially kind of looks at a specific binding area of the receptor. And what we found was the venom actually targeted this area. And now what's interesting is that venoms that usually target these receptors are usually predatory venoms because they want to cause paralysis to their prey item, right? So easy to immobilize and easy to consume. There's less of a fight there. However, it was strange because we know the stonefish venom is a defensive venom. So why would this venom want to cause paralysis? And so we kind of hypothesized that a lot of predators of stonefish, or particularly earlier relatives of stonefish, were things like very large sharks and chondrichthyes, so rays, for example. And so we hypothesized that if the venom could kind of paralyze the inside of the mouth or the mouth area of these sharks, which, by the way, sharks and rays, for example, their tactile sensors are all around that area, so they basically feel with their mouths. So if you could kind of paralyze this area, this means that the shark can no longer be as effective as a predator, right? Using its mouth area. And so this is one hypothesis we came up with. This is just probably the most parsimonious um, hypothesis we could have came up, but it kind of makes sense in the grand scheme of things. And so what was interesting there is that we found that a seemingly predatory function of a venom was we found it in a defensive fish. And so we wanted to actually take that further. And so we looked at some of the clinical records of what stonefish actually cause. And again, what happens is when someone is stung by a stonefish or any defensive venom, for example, they have arrhythmia, so changes a bit in the heart rate, so a very increased rate and then a slowed down rate. And so we looked at the literature again, and it was kind of inconclusive what area these venoms were targeting. So without getting into the kind of pharmacological side, there was two kind of receptors that it could have been. It could have been calcium channel receptors or beta adrenoreceptors. And so I tested the venom across a new assay that I'd been kind of working on and developing, which was the calcium 1.2 channel. And again, we found that the venom actually targeted this area. So we concluded that this is definitely uh, a new target for this venom. And then thirdly, we wanted to look at kind of the blood side of things, how the venom affected the blood. And so what we found was that it caused anticoagulant activity, which for a defensive venom, that really makes sense. Because if anticoagulant kind of allows for other painful toxins to get within the system. Yeah, and so we found actually anticoagulation. So we found these three kind of new and confirmed these three new pathophysiological functions. And now what's more interesting is that what we took another approach to this. We tested these same assays with venom that was fresh, so taken straight from the fish and then used on these assays. And then secondly, we lyophilize the venom. So for anyone who doesn't know what that is kind of you basically dry down the venom to a powder form, and then you can kind of store it away and things like this, and bring it back up for other purposes and other uses. And so what was interesting is we found that the fresh venom worked on all these assays perfectly fine. But if we lyophilize the venom, a lot of these activities dissipated or just wasn't present at all. And this is likely due to the fact that kind of the toxins that are causing these activities are very large molecules and they break down very simply under lab conditions. And so this might, I very much highly doubt it, but in terms of looking at antivenom for the stonefish, they usually create the antivenom using lyophilized venom. And so it's possibly that the antivenom, although it works very well, and I'm not saying it doesn't, it will definitely help a lot. It might miss a few of these toxins that are kind of broken down through the lyophilization process. And so that was another thing that we kind of pointed out, and that we've had a lot of companies, speak about and these kind of things. But yeah, just as a disclaimer, like the anti-venom works perfectly fine. So yeah, I just don't want to say like, that it doesn't work. It works perfectly fine, particularly yeah, the the purposes of the very large lethal toxins, although not lethal to humans. Um, but the ones that cause extreme pain are pretty well kind of
And so
neutralized within the antivenom.
okay, imagine if there is an antivenom specifically produced against stonefish, stonefish envenomation, I would say that there is at least like a good reason to make it in the first place, so that the stonefish envenomation must be a reason for serious clinical concerns. So not only for stonefish, like what is the clinical relevance of fish venoms compared to, for instance, other terrestrial more famous venomous animals?
Mm. Okay, yeah. So So obviously the first protocol is intense pain at the local site. Um, so you have this extreme pain with a lot of these venoms, which is the most for humans anyway, particularly is the worst part. Um, but what can happen is you can also get kind of cytotoxicity at the local site. So you get kind of necrosis around the wound. In a lot of cases, this doesn't turn out to be kind of a systemic necrosis where you see, you see things like in a lot of viper bites where you have like whole limbs kind of just absolutely destroyed with cytotoxicity. It's not this bad in fish venoms, thankfully. So the first protocol is kind of trying to get rid of that pain in the clinical manifestations. Long-term sequelae can exist though. So for example, there's been reports of stonefish envenomations where they've had compartmental syndrome, and things like this. So like long term effects where limbs just don't kind of function properly anymore, and so that I think, but in most cases that compartmental syndrome is kind of the worst case scenario. So if you can actually kind of stem the pain and the venom and get some hospitalization very quickly, then these long-term sequelae don't really manifest. These are like kind of worst case scenarios where you're in the middle of nowhere, you've been stung, and you have to ride out the pain with no medical professional help. But they can exist. And I was actually contacted in November last year by a lady over in the US who had been stung by uh, Scorpion guttata, I think it was, which is a scorpion fish, and she had long term effects. She had, yeah, she couldn't move her limbs properly. She kept getting kind of lack of sensation all throughout the limbs, and this was a year after the sting. Yeah, so you do get these long term manifestations of things that could potentially put someone out of work or anything like that. So yeah, so a lot of people think of fish venom as just painful, but there are instances where they can be detrimental to people in the long term. But as you say, they're not as clinically relevant as snakes, for example, which are a huge burden. Um, yeah, and I can talk a bit about the basically also kind of if you are stung by a fish, a venomous fish, what the protocol should be
Oh yeah,
to
no, that would be
kind of
very interesting.
yeah.
Yeah.
Okay, so yeah, so if you're stung by a fish, it's recommended that the first port of call to do is something called hot water immersion, which is basically you want to submerge the affected limb in hot water, water that's not hot enough to scald you, but hot enough so you can tolerate it. Okay, and so there's many different theories of how this works, and it's very debatable. But what we do know is that in a few cases, and in quite a few cases, pain subsides depending on how quick you immerse the limb within the hot water. Okay. It doesn't always work though, and that's something that people need to be wary of. So hot water immersion does work sometimes, and quite a few of the times it does, depending on the severity of the sting. However, it doesn't work all the time. And so if it doesn't work or the pain doesn't start to subside after hot water immersion, your next protocol is obviously is things like what the medical professionals can give you. So it's like morphine or anything that can actually kind of control that pain for you because it is very, very bad. And then actually, in fact, the anti-venom is usually the last port of call for the medical professionals. That's kind of their last resort because they most of the time just want to stem the pain. Um, yeah, and so obviously the first protocol is called the medical professionals. But then there are a lot of other things. So for example, very young people and very old people, particularly in old people who have potential heart problems and things like that, things like this, that can be really bad for them because we know that a lot of these fish venoms actually, um, you know, kind of play with the heart rate and affect the heart in a way. So if you have a weak heart, this could potentially be very bad for you also. But yeah, so the first protocol is kind of that hot water immersion, which if you're stung at a beach, that's got lifeguards, for example, um, they should be aware of kind of how to treat that, particularly here in Australia.
Oh yeah, that makes sense. Since I mean, of course, it had to be Australia again. Like
It's always Australia.
any sort of venomous stuff. I mean, why do I even bother asking? So, but so yeah, you were saying that yeah, venom evolved 19 times independently in fish, and so I was
It could
wondering,
be
what
more. It
about
could
like
even be more. It could be more times too.
yeah,
Could even be
19
more.
times that we
It could be
know of.
more.
Yeah, but
Yeah.
good point. Thank you. So I was wondering how about like what is the diversity of venom delivery systems in fish? Because like you
Mm,
mentioned spines, which seem to be the most common, if I'm not mistaken. But then you also have fang blennies, and you have I think so, at least some fish that have that inject venom from their tail or something. If I'm not like
mm,
some stingrays that do that. So how do fish actually produce and store their venom?
yeah. Okay. So yeah. I mean, so as you said, as it's evolved on 19 different occasions, there's a multitude of different ways that these have evolved. So if we just look at the ray fin fishes, for example, you have the venom that can be on the dorsal spine, it can be on the opercular spine, it can be on the caudal spine, it can be on the collateral spines, which you find in some toadfish. And if you're a lionfish, you have basically them on every single one of them. So there's what like four different spines that the venom can evolve on in just the ray fins, for example. Then you have your fangs and your fang blennies. You also have fangs in the monognathid eels, so your jaws eels they're called. You have kind of these in the lampreys. For example, you have they're not necessarily fangs; they're more like a kind of chitinous plate of protruding
Mm.
teeth. They're really you can Google what their mouths look like, but they have this, and then you
Oh
have
yeah,
this
that
like
yeah.
a
To
radulate at the back.
everyone listening, yeah, please, yeah, Google what lampreys look like. It's the stuff of nightmares, but I love them. They're so cool.
And then you have the serrated barbs of stingrays, for example, which are actually really cool because of their serrated edge. They have kind of a backwards, an antro retro kind of serrated edge, and so once the spine actually injects or is penetrated in to an organism, pulling it out makes it a lot worse because of these kind of backward facing spines, and so you get more mechanical damage kind of with the removal of. And there's images on Google where there's kind of like I think it's like dolphins and whales, and they just have stingray spines hanging out of them because obviously once they're in, it's very difficult to pull out, and you don't want to pull them out. And that's particularly evident with human envenomations too, is that you get a lot of mechanical damage actually trying to pull them out. And there's quite a few papers of medical professionals actually kind of trying to work out the best way to actually remove the spines without causing a lot of surgical damage or too much scarring for the person. And yeah, so they're just kind of uh, a broad sense of the actual mechanics of what the venom systems can attach to. And so in terms of the venom systems you can have within fish, there's usually two. There's one where the venom is actually contained within what's called holocrine cells. So these are cells that release the venom when they are ruptured. So this is the majority of ray fin fishes, for example, that use the dorsal spine, is they have a kind of a sheath that's surround. If I use this pen as a spine, they have a sheath that's kind of surrounding the spine, which within this sheath there's holocrine cells. And then once the someone penetrates the spine and it goes in, these cells are ruptured and the venom kind of releases into the system. But then you have a more I guess more advanced system in the stonefish where you actually have these glands that are either side of the spines, these highly kind of evolved glands with a venom duct. And this is actually the gland system within fish is very rare, which suggests to me that the holocrine system, where the integumentary sheath is just kind of pulled away and the cells are ruptured and the venom is released, is adequate enough for the majority of fish. And so they don't kind of need to evolve this gland system that contains. So it's like a gland system similar to what you would find in the glands of snakes, for example. And it's really interesting because the real question is what selection pressure was so high that made the stonefish had to evolve this, this more advanced system, that's a really fascinating question for me. And then similarly with the fang blennies, for example, they also have similar kind of these kind of holocrine cells that when the fangs puncture, the venom is kind of pushed through a groove on the tooth and it kind of goes into the system. Yeah. So they're kind of the main venom systems within fishes. And I'm pretty certain we'll probably discover a few more as the years go by.
Yeah, at this point, I don't even know what else to expect.
Expect the unexpected. There's that many fish, and yeah, just
And yeah, but I'm glad you mentioned selection pressure in regarding the stonefish because it reminded me that yeah, a couple of months ago I went to the aquarium here in Copenhagen and they have one tank dedicated to venomous and poisonous fish. That's why they also have a
mm,
puffer fish in there, which is poisonous,
yeah,
of course, not venomous.
yeah,
But yeah, they also had lionfishes and reef stonefish. And I mean,
yeah,
when I looked at those two, it was like two completely different worlds because like the lionfishes are so beautiful and brightly colored with all these long flashing spines and that almost look like feathers. And then you go
mm,
to you look at the reef stonefish, and I mean, in that moment I thought that probably the people who first described it decided to name stonefish because third fish was not acceptable in scientific terms. Because I mean, honestly, that's exactly what it looked like.
yeah, yeah. And you wouldn't. I mean, if you were just stumbling along the rocky shore, you would not be able to see that. No, you would never be able to. I mean, it has probably one of the best camouflage systems I've seen. It is perfect.
Yeah,
We
no,
had one
exactly. And
in
so,
sorry. Go on.
oh yeah, no, so yeah, that's what definitely struck with me, which is the contrast between yeah the lionfish, which is basically like wants to be seen by everything, just so that everything knows not to mess with it, and
Yeah,
then the stonefish which does the exact opposite, but again for basically the same reason. So they have an extremely painful venom that will make anything that messes with them regret it sorely.
yeah,
So
yeah.
yeah, in
So
terms of like
what's actually interesting? What's actually interesting where you mentioned there about the complete difference in these two fish, but when you think about it, these are actually in the same family. These are both in the Scorpaeniformes family, right? So they're actually fairly closely related, but yet their venom systems are kind of convergently different, and even obviously the look of them, right? They're very different in terms of how they look. But what's actually more interesting is the fact that we have these convergent evolutions of the venom systems, okay, even within closely related species and within families. But what's interesting is their main toxins, the quote unquote stones toxins, for example, they're actually very closely related when you plot them out on phylogeny. And so what's interesting is that there's clearly whatever these venom systems are evolving from, um, it's something that all fish kind of need and use. So the main hypothesis right now is that a lot of these venoms and toxins evolve from what we call ichthyocrinotoxins, which are these secretions on the skin layer of fish that kind of can help with to reduce parasites, can act as antimicrobial compounds on the skin, and also in some cases can act as kind of a noxious anti-predatory purpose. And so the main theory right now is that these ichthyocrinotoxins have somehow been kind of recruited into the venom systems of these fish. And so, and that's a really important thing to note is that we have these 19 separate convergent occasions of venom systems evolving, but the venom toxins between all these different convergent evolutions are very similar. Um, I guess once you start moving out towards kind of things like your fang blennies, and also kind of stingrays, for example, you do get differences within the whole venom composition. But the main lethal toxin, the stoners type toxins, which are pore forming toxins, they actually are very highly related. So clearly, there's something that these evolving from that's within all fish, right? And all fish kind of utilize, I guess, the genes of whatever the precursor to these stoneless toxins are, which is really fascinating area to kind of explore.
Oh yeah, it definitely sounds like there's a lot of room for very interesting research in
Mm,
there. But so yeah, that brings me back to the very beginning of this of our chat, because we were saying again that fish venom is definitely not a majorly known topic in among the general public, but even in terms of research, it definitely hasn't received nearly as much attention as venoms from terrestrial animals, I presume. And so, do you think that is only because of yeah, the obstacles represented by the sea, so that you actually have to go to dive down and see these
mm.
to get these animals, or is there are there also other reasons why fish venoms don't have as much appeal as even non-lethal terrestrial venoms?
Mm. Yeah, so I think the first main reason is, as you've just said, is that they're not as clinically relevant than things like snakes and scorpions and spiders, for example. Another main barrier is, again, as you aptly mentioned, was the fact that you can go and capture snakes. I could go an hour outside Brisbane where I'm living and and potentially catch some very very dangerous
Yeah.
um, highly venomous snakes to look at. Whereas these stonefish or any fish, for example, they're difficult to capture. Um, they're also difficult to maintain within aquarium systems. Not a lot of universities or research centers have the facilities to actually keep these organisms to kind of continuously milk and continuously look at their venom. Um, and then I think, to my mind, I think one of the main reasons is the fact that a lot of the toxins within the venoms of these species are highly labile, so they kind of just really degrade under basic lab conditions. Like a lot of snake venoms, for example, you can dry them down, you can lyophilize them, you can do a lot of things to them, and they'll still be highly efficiently. The toxins will be efficiently working once you come back to do the relevant experiments on them. With fish venoms, for example, they can break down under the basic lab conditions. So one freeze thaw cycle, and the main function can be degraded. And so I wrote quite a bit on this, like kind of the best way to try and get fish venom research back into the mainstream of toxinology. And so there's a few kind of protocols that you can kind of take, and again, there's still a lot of barriers, but I think we do need to look at the venoms of fish. I think it's a huge area where we're missing, and I think by understanding the fish venom system, we can actually apply that to other venomous systems of the animal kingdom. And so, yeah, I think the main reason why a lot of people kind of stemmed away from working on fish venoms is the extreme liability of their venom and toxins. It's very difficult biochemically to kind of keep under control. But isn't that what science is about? Let's figure out a way to kind of you know stem that kind of degradation of the venom, and let's keep working at it. And that's kind of where I see my area of research in the future.
Oh yeah, and I mean, as long as as long as the situation stays like this, then you will be able to remain one of the world's top experts in in fish venom for quite a while, it
Yeah,
seems.
that's good. Yeah. Maybe it's a foolish dream. I don't know. Maybe we shall see.
And yeah, I also forgot to mention it before. If anyone listening has any questions for Richard, just drop them in the chat, and we will readily see them. And on my account, I have one left that I'm definitely very curious about, because of course you've been working with the venoms of these animals, and you also mentioned at some point before that you also personally extracted venom from
Mm.
fish, at least on some occasions. I remember seeing the pictures of that time you extracted venom from this that giant big
So
stonefish that
yeah.
you had brought to the lab, and I was like, how do you even get venom out of a fish in a way that allows you to use it for research? So how does
Yeah,
that
so,
work?
so yeah, the stonefish is actually probably the one of the most easy ones because it has that venom gland system. Um, so yeah, so basically there's two ways you can extract venom from stonefish. You can kind of push down the sheath around the spines, and what happens is the venom glands kind of collapse in on each other, and the venom can actually spray out. There's videos on YouTube of I think it's Jamie Seymour at his lab up at GCU, and he's actually showing how far the venom can actually spray out of the spines. So it actually the venom gland, the pressure in the venom gland system actually just pushes and it can actually spray. So you can actually extract that way, or you can do it, which I'm not a huge fan of this method. Because I think it's a bit more invasive, is you can actually put a syringe because you can actually see the glands because they're bulbous, you can see it underneath the integumentary sheath. So you can actually kind of pinpoint where the gland is and kind of use a syringe to extract that venom. So that's two ways you can do it with a fish that has a venom gland, a strict venom gland system. But for the majority of other fish, so for example, I've milked the weaver fish, both the lesser and the greater over in the UK, and they have the holocrine system. And these are a lot much easier actually because you can just use a Eppendorf tube with kind of a sponge at the tip, and you can place this over the spine, and then the venom is absorbed into the sponge, and then you can do centrifuge and things like this. So I think the holocrine systems are a lot more, a lot easier. But the hardest part is obviously once you take the fish out of water, they don't kind of want to stay still. So they're kind of,
Yeah, I mean, I can
you know,
honestly, I don't. I can we really blame them? Like,
I know, right? Yeah, so they can be, but you give them a minute or two and they kind of settle down. But obviously working with fish out of water is a lot more dangerous for the fish than it is for us in a sense, right? Because they need water to survive. So yeah, so time is of the limit also in that sense, kind of, and obviously you don't want to stress out the animal as much. So you want to kind of keep that stress to a minimum, and so yeah, I mean it's not the easiest technique, but it's a lot less dangerous than it is milking snakes, to say the least. Yeah.
and
So,
so
and
they,
also, something for example, like a stingray, their barbs actually displace, so you can actually take the barbs from them. I've had a lot of studies work on taking the barbs from stingrays that are actually probably already recently died, which is probably the best method. Not to kill them for this purposes, but for other research lines. But you can just simply extract the spines of them anyway, and so that's a fairly straightforward method too.
oh yeah, and then David has quite an interesting question in the chat. So, do any fish sequester venom from other organisms rather than producing it themselves?
So yeah and no. The straight answer is no. I'm actually not. So usually, if you sequester toxins from other organisms, it would usually be distributed as a poison rather than a venom. As far as I'm aware, I don't think. I mean, Lorenzo, you might know of an example where venom can actually be, I guess, sequestered or bioaccumulated. I'm not sure, but as far as I'm aware, no. The majority of venom systems are produced themselves, so they produce the toxins from within their own cells. And I guess that's probably one main definition of the use of venom, whereas a poison is a bit more, um, I guess, malleable in terms of how they can acquire them toxins through bioaccumulation or uh, you know synthesis and sequestration themselves. So yeah, it's a good question, David, but yeah,
Yeah,
I don't think so, no.
probably. David, correct me if I'm wrong, but I think that the reasoning behind the question was influenced by pufferfishes, which indeed
Mm,
sequester tetrodotoxin, which is their main toxic
yeah.
compound
I guess, I mean,
from their diet. But
I, I guess thinking about it, yeah, I mean, it's not, yeah, I guess. So I guess I think it boils down to the main definition of what you define as a venom versus a poison,
yeah,
right? So I mean, if you, I guess in a sense, yeah, um, you could sequester toxins from other organisms, and kind of have that layered over the spines. And then once the spine is punctured, then I guess it's technically classed as a venom, right? Particularly if it's used um, in an ecological relevance, right? So yeah, actually going back to the question, I can see that it could potentially evolve actually, but I don't know any instances of it. Sorry, David.
Oh yeah, until
So
until
yeah,
until until someone finds it, because
yeah,
like you said with fish, I mean, I wouldn't rule it out in principle, indeed.
yeah. Oh no, definitely not. Definitely not. Yeah, definitely not.
All right, so I since there are no other questions from the audience, I would say that concludes our interview. So once again, Richard, thank you so much for coming here. It was extremely interesting, and honestly, I really learned a lot. There are many things that I knew nothing about, and it was great to see you again, of course.
Yeah,
Uh,
same. It's been a pleasure. It's been a pleasure. Anytime. Happy to
Oh yeah, and
do this
so
anytime.
the interview will remain available here on Habs and also on my personal channels on Facebook and YouTube. So if you want to remain updated on new episodes, which I already have a few planned, then subscribe to basically each and every one of them. And indeed, stay tuned because there will be very many more exciting talks, exciting interviews in the future. So in the meantime, I wish everyone a great weekend, and I hope to see you soon for the next episode of the Snake Man's Lair. Thank you again.
Cheers. Thank you.